So hello everyone. Let's start our third and last session of the day. I hope you have been enjoying so far. So the second part and the final will showcase innovative startups in UK. And we are going to have Dr. John Maher, Chief Scientific Officer at Leucid Bio, and Dr. Emily Warner, Senior Scientist at Icarovec. Each speaker will have 10 minutes to present, and the questions will be taken afterwards in a panel discussion. And for last, but not least, we are delighted to have our keynote speaker, Professor Moin Selem. He's professor at the University of Bristol and founder of PurSpring Therapeutics. And we'll finalize the day, as I said, with the last panel discussion with all the company's representatives. Thus, please write down your questions for the end, and they will be answered at the end. So, our next speaker is Dr. John Maher. He is a clinical immunologist who leads the CAR Mechanics Research Group within King's College London. He played a key role in the early development of second-generation CD28 CAR technology while a visiting fellow at Memorial Sloan Kettering Cancer Center, an approach that has achieved clinical impact in hematological malignancies. His research group is focused on the development of adaptive immunotherapy using CAR-engineered and gamma-delta T cells with a primary emphasis on solid tumor types. He's also the scientific founder and chief scientific officer of a spinout company named Leucid Bio. In addition, he's a consultant immunologist within King's Health Partners and Eastbourne Hospital. Now let's welcome Dr. John with the presentation titled Parallel CAR T Cell Immunotherapy of Solid Tumors. So please.
Okay, thank you very much for the introduction, and thanks for the invitation to come and speak. I can't remember the last time I did a 10-minute talk, so this is going to be interesting, but I promise to try to keep on time, and these are some disclosures. So I had hoped to tell you a little bit about a clinical trial that we're running, but I've removed that from the presentation in the interests of time. So instead, what I'm going to talk to you about is the kind of core technology that we developed some years ago, which led us to spin out this company called Leucid Bio from King's College London. So I imagine that everybody here in the room is familiar with the different generations of chimeric antigen receptors which have been described, going right back to 1987, the first description of a CAR by a group in Japan, Kuana et al., rather, a paper that's not commonly cited, actually. But the difficulty with these first-generation CARs, where you have only an activating domain on the inside of the receptor, is that when they were tested in the clinic, they simply weren't enabled. What's caused all the hoopla around CAR T is this beast here, the second-generation CAR, where you introduce a single costimulatory module upstream of that activating moiety. And this is the technology which has made the leap in the context of blood cancers. Somewhat surprisingly, however, when we've moved from second to third generation CARs, where you introduce a second co-stimulatory module into this structure, we haven't seen any further real improvement in clinical impact. And this is surprising, really, because in this architecture, you can introduce two very different types of co-stimulatory molecules, such as CD28 and 4-1BB, for example. And these individually are both very effective in the second-gen platform. And we know that these different flavors of co-stimulation can synergize in naturally occurring immune responses. So that led us to speculate that maybe there's a problem with this kind of linear fusion design. Perhaps these co-stimulatory moieties need to be situated in their natural location in proximity to the plasma membrane in order for them to work. We have known that that's the case for CD28 for more than 20 years, so why shouldn't it be the case for molecules such as 4-1BB? So because of that, we designed this lateral structure, which we refer to as a parallel CAR, where, for example, we co-express a second-gen CAR with CD28 here, alongside a chimeric co-stimulatory receptor, which is a different co-stimulatory unit, such as 4-1BB. So with that in mind, we have developed at Leucid quite a lot of different pCARs, but in the interest of time, I'll just tell you about one family of pCARs which we have developed and which is targeted against this group of ligands, the so-called NKG2D ligands. Once again, many of you in the room will be familiar with NKG2D. It's a receptor expressed by natural killer cells and some T cells, and it patrols for these 8 different NKG2D ligands because these ligands are a marker of cell stress. So this is an innate immune surveillance mechanism looking for cells which are potentially dangerous to us. And we thought this could be an interesting set of targets for tumour immunotherapy. And we're not unique in that respect because there is a company, for example, called Celiad Oncology who have quite an advanced NKG2D CAR-T program in the clinic in multiple clinical trials at the moment. Now, this movie here is an example of what happens when you test these NKG2D pCAR T cells in an in vitro tumour model. And I should point out that what we've done here is to mix tumour cells alongside mesenchymal stromal cells to kind of simulate in a very simplistic way the microenvironment that's found in a tumour. Many solid tumours, such as pancreatic cancer, for example, have a dense stromal infiltrate. And these stromal cells actually provide the bulk of the structure of the tumour. And one of the things that they do is they produce chemokines, which naturally attract T cells into the so-called stromal highway, as it were. And when T cells are sitting in the stroma, I guess what they are not doing is attacking the tumour. So it's a kind of a defense mechanism that tumours can often use. The difficulty, however, from the point of view of NKG2D ligands, is that stromal cells are also stressed within the tumour, and consequently, if you could maybe just start the movie for me, please. If you add on these T cells, you'll see that the first place that they go to is the stroma. They start attacking the stroma. The tumour cell islands begin to coalesce, almost as though they're trying to escape from the T cells. But as you can see, then the T cells turn their attention to the tumour cells and destroy them as well. And you can also carry out these kinds of models, if you could run this movie, please, in a 3-dimensional spheroidal structure. So what we have here is a ball of cells where the tumour cells are labelled with GFP, the stromal cells are labelled with RFP. It takes a few hours for the T cells, the CAR T cells, to break their way into this core of cells. But as you can see by now in the movie, it's becoming progressively more and more homogeneous as the T cells infiltrate and destroy both the tumour and the stromal cells that are present. Now you can even see this actually with the naked eye, because what you see here are 5 tumour spheroids which have been co-cultivated with untransduced T cells, and you can retrieve them more or less intact. And if you deaggregate these spheroids and put them through a flow cytometer, you can see that there are lots of tumour and stromal cells present here. There are some T cells as well, but notice how small the T cells are because, of course, they have not been activated in this culture. With a linear CAR, again these spheroids are visible with the naked eye, although if you deaggregate, you can see that the proportion of tumour has been somewhat reduced, and these T cells are more activated, indicated by the larger scatter that they achieve in flow cytometer. Whereas with the pCAR, nothing is visible with the naked eye, and if you deaggregate what you can't see, as it were, you can see that the tumour and stroma is completely destroyed, and you've got highly activated T cells present. And then if you move that in vivo, here you can see 2 examples of xenograft models in which we have treated NSG mice with an established tumour burden with our NKG2D pCAR T cells. Now the control in each case is this mysteriously labelled 2G CAR, and in actual fact what this is a pan-ERB CAR. It's the CAR that we have in the clinic at the moment. If we take those 2G CAR T cells and expose them to these pancreatic tumour cells in vitro, they will recognise them and destroy them. But in vivo, they have no therapeutic activity, whereas by contrast, the pCAR T cells cause complete eradication of the tumour, and a re-challenge with that tumour is rejected as far out as day 88. It's a very similar story here with this mesothelioma xenograft, with the exception of the fact that the 2G CAR does have a degree of anti-tumour activity in this model. And here's a third example. This is a mesothelioma PDX, which we have established from a patient at Guy's Hospital. And you can see that this tumour was growing for over 100 days in the mice before we treat intravenously with these pCAR T cells, causing regression of those tumours, in contrast to all of the controls, which have no therapeutic activity in this model. So I think this is my conclusion slide. It's like buying a house. When you design cores, it's all about the location of these units in the fusion receptor. And I'll finish by thanking, by thanking the many people who have carried out this work, both in my academic group, known as the Core Mechanics Group. I didn't invent that name, by the way, and also our company, Leucid Bio. And I look forward to questions at the end of the session.